Anthropology series. So today we'll be completing this 9.4. We were doing genetic counseling, and we had studied about genetic screening and all of that. So the last topic would be DNA fingerprinting. So this word DNA fingerprinting means that see what basically it is different from the idea that we have in our minds about maybe it's something about the fingerprints, because the name is DNA fingerprinting, but it is not true. We are not here studying about the fingerprints as such, but we are trying to understand the DNA, and you can say DNA, we are profiling the DNA. That means every human, we have talked about that every human has a unique DNA, right? But it is not entirely true also, because 99.8, or you can say 99.9% of each person's DNA is same. Okay, so there is a 0.1% difference, and अगर ये 0.1% भी same होता, तो we would have been like maybe some animals or maybe other individuals जो they look alike, right? Who look alike. So that you can say the genetic polymorphism that is there in us, right? So these are the little things that have made us look different from each other. But having said so, we are saying that 99.9% of our genetic makeup is same, and there is 0.1% difference. But this 0.1%, if you see, we have, like, you can say trillions of cells or DNA in our body. So even 0.1% be, it will be a lot of number, right? So when we study about DNA fingerprinting, what we are trying to do, we are trying to understand the profile or the print of our DNA, right? So basically क्या होता है, and it can be collected from any part of our body. You can take it from the even the fingerprints on your hands, right? You can take it from your hair sample, right? So DNA collect करना है, and then we have to look about it. So now let's understand what the whole idea is all about. Yeah. So DNA profiling, or it is also known as, you can say DNA profiling, you can say DNA fingerprinting. DNA fingerprinting थोड़ा सा misnomer है, because it's not talking about the fingerprints of our hands, right? So just keep that clear in your mind. It is nothing to do with the fingerprints of our hands. So it is also known as DNA fingerprinting. This is the process to determine an individual's DNA characteristics. We have talked about it. People have 99.9% similar DNAs, but there is 0.1% DNA that is different, and which is and much part of our genes are inherited from our parents. So thereby iska application, this is one of the most, you can say, revolutionary studies or revolutionary technologies in the field of genetics, because इसको हम use करते हैं in finding out criminal suspects, right? In paternity issues, property disputes, any rape cases. If some female gets pregnant after a rape scenario, so who's the father, to determine who, so all these medico-legal cases, can be solved with the help of DNA fingerprinting. So ये तो application होगी, where we can use it in the criminal suspecting, paternity dispute, property dispute, rape cases, in fact murder cases, to find out who was the person who was murdered, right? So murder cases, many medical legal cases, we can use it, and it can be also used in the study or fields of zoology, botany, agriculture, all of that. ठीक है? So this is what you understand कि DNA fingerprinting क्या है. We are trying to find out, every person has a unique DNA characteristics, but somewhere down the line your DNA matches with your parents' DNA, right? So to find out, एक तो हो गया we are trying to find out DNA profiling हम कर रहे हैं to know your ancestry, ठीक है? In the cases of trying to paternity dispute में, we are trying to see who is your parent. In some cases to know your identity कैसे? Because for example, there's a government, okay, they have your biometrics, they have similarly your fingerprints, right? Someday you go missing, okay? So these fingerprints that they have taken, what is this? This is your part of your DNA. So they can, with the help of this DNA, they can find you, right? So this is to use to find the identity. Similarly, if you're murdered, say somebody, and you're gone missing, so government, if they have this kind of a data, or they find certain things, for example, you're murdered and left your DNA here and there, so they, with the help of the DNA, they can find you. So every person has a unique DNA which does not matches with any other person until and unless you are identical twins, right? Because हमने बात किया था, because they are made up of one ova and one sperm, so that is why they have their DNA is by and large similar. But उसके अलावा, even with your parents, your DNA is not the same. Obviously it has certain characteristics that would match, but ultimately 100% similarity नहीं होगा. So that is why it could also be used to identify. So क्या है DNA profiling? It is a process of determining an individual's DNA characteristics. ठीक है? So we have talked about that although 99.9% of human DNA are the same in every person, but there is 0.1% that makes us different unless we are monozygotic or identical twins, जिनके केस में DNA is the similar. So DNA profiling, now how to do it? Because अभी हमको समझ आ गया, it has a lot of advantage. जैसे if I have to tell you certain famous cases, you know, where this DNA profiling has been used, so you can say the 9/11 attacks. ठीक है? So it was used, वहाँ पे there were like more than 2,000 people killed there, right? So who died in the attacks? To make sure we are recognizing the same people, वहाँ पे use हुआ. Similarly in India, you can say the 2013 Uttarakhand floods happened, and to see who all were the people who were the victims of that flood, to see वहाँ पे use हुआ. Similarly, these things have ethical concerns also. What ethical concerns? For example, if government has your data, they can track your data, right? So in many cases, जैसे इसका बड़ा एक infamous case हुआ था, Cambridge Analytica case, right? Cambridge Analytica case. इसमें क्या हुआ था? Questions arose about DNA information use and data mining. So basically हम भी बोलते हैं ना, many times in the cases of Aadhaar भी ये criticism आता that government has our data, they have our fingerprints, they have our, so much of data is there, right? So if this data gets leaked to any person, ठीक है, तो क्या होगा? Because this is too sensitive data, this is personal data. Similarly when NCRB, National Crime Records Bureau, ठीक है, उनके पास जब when there are criminals that go to them because of some crime that they have committed, they have their DNA profiles, right? That means they have their full DNA. So कल को कभी भी कोई और murder होगा, कोई crime होगा, the NCRB can use your DNA fingerprint to check whether it was you or not, right? So these are the ethical concerns also which can occur when there is a breach of data privacy or your data leak is there. So these are certain cases that can come with it. So ये समझ गए? Then now the point is, हमने समझ लिया what are the ethical concerns, where it has been used, what is DNA fingerprinting, and why is it so important in today's day and age, right? But now how to do it? Now DNA profile uses repetitive sequences that are highly variable, called variable number tandem repeats. In particular, short tandem repeats, also known as microsatellites and mini satellites. Now these are similar between closely related individuals, but so variable that unrelated individuals are unlikely to have the same VNTR. Now I'm sure you did not understand, so don't worry, I'll take you very slowly to the whole concept. This is a DNA. Now just understand, देखो DNA जो होता है ना, it is made up of these four, you can say these are the backbone, or these are the sugar phosphates which make the whole DNA, and these are adenine, thymine, guanine and cytosine. ठीक है? Now you can see that they are paired in a group of two and two, right? So what does this mean? This means that these are sugar phosphates. हमने 4 बोले, adenine, thymine, guanine and cytosine. So always क्या होता है, adenine will pair with thymine and guanine will pair with cytosine. This is the thing. That is why they will always be shown together. Always be shown together. ठीक है? Always be shown together. Similarly, adenine and thymine also shown together. So this is the thing. ये आपको just for your understanding, because I'll be using these things to make you understand what is variable number tandem repeats, because that is the topic we need to understand. ठीक है? So you understood this is a DNA. It goes on. It's a double helical structure like that. ठीक है? And it is made up of these sugar phosphates, which are these 4, and who combines with who. So ये समझ गए? This is the DNA. You have understood part of the DNA. Now what happens? Just see. For example, मैं एक coding कर देती हूँ, because ये coding है. Because every person has a unique coding. Okay. So for example मैं बोलूँ there's a okay. Now हमने बोला कि it is a double helical structure. ठीक है? जब when we study the DNA profile, we need to single out these double helicals. So we take them, we destrand them. ठीक है? ये पहले ऐसे है ना, हमको पहले इनको अलग करना पड़ेगा. So we do that, separate them. ठीक है? We'll see the whole procedure also. Now I take a part of this. ठीक है? For example, I take this part. Now this part has a coding on it. ठीक है? Now let me make a coding. Adenine, thymine, guanine, cytosine, thymine. Okay. So let me make this as a tandem repeat. Okay. Now this is not a repeat. Repeat कब होगा? When I'll make more of it. ठीक है? Now I need some space here. So you understand what we are doing. Basically we are trying to, okay, now it can be coding कुछ भी हो सकता है. It is not like adenine, thymine, guanine, जैसे मर्जी हो सकता है. Guanine, फिर cytosine. Now let me make this as 1. Now let me make more of it. Same. I'm not changing it, guys. Okay. A, T, G, C. Okay, sorry. G, C, C. A, T, G, G, C, C. A, T, G, G, C, C. Now this becomes a variable number tandem repeat. Okay, these are tandems. They become and they are variable. That means इसके आगे कुछ और sequence हो सकता है, इसके पीछे कुछ और sequence हो सकता है. They are variable, but here till here it is a sequence that is repetitive. This is a repetitive sequence. So बीच में in the variable sequences यहाँ पर there could be some other either thymine, guanine, cytosine ऐसे हो सकता है. यहाँ पे adenine, cytosine, thymine हो सकता है, right? So in a sequence, जैसे हमने बोला this was the helix, we took this part and in this part this coding is there. ये पूरा इसमें line में चल रहा है, right? So this is the coding where some part is repeating itself. So this is the variable number tandem repeat, and इसमें इस case में it is 4 times, right? 4 times. So let us say this is the mother who has this unique coding on her DNA. Similarly, there will be a coding on the father also. So let us say कि father के coding में there is three times a variable tandem repeat is there. ठीक है? Variable number tandem repeat is there 3 times. Somewhere, right? Now this both will arrange itself in the baby, because obviously it will many times these sequences go in the children, right? We have talked about that, कि some part of our genetics is inherited from our parents. So this might be present in the child. With the help of this, for example, if I give you a now different picture, because हमको थोड़ा we will make it more clearer, right? Let us say this is the mother, this is the father, and this is the child. Now I am just taking a sequence of the mother, the child and the father. let us say the father had these tandem repeats here, dot वाले, ठीक है, this is the dotted वाला father का है, 4 time repeat है, ठीक है, या whatever you can say, this is just a repeat in the father's DNA. Mother का it is something like this. This is extra guys, the yellow part that is coming out, ये थोड़ा extra है, I'm not making it with a lot of precision. Now the child might have something like this, okay, the child could have something like this. So you can see clearly the child has inherited some part from the mother and some part from the father. Now this is the variable number tandem repeats that has inherited that the child has inherited from their parents, ठीक है. So this is the variable number tandem repeats also I've just shown you. These are certain, you can see these are the sequences that are repeating itself, ठीक है. In case इसमें दो बार repeat हो रही है तो 2 repeats, ये 3 repeats का sequence है, then there could be 4 repeats, 6 repeats, ठीक है, different sequences can be there. Now variable number tandem repeats, short tandem repeat same but shorter. Now just ignore this, ये मैं आपको समझा दूंगी. Okay, so this you've understood. Now this is the gel electrophoresis. This is how you actually in this machine you put the VNTRs, ठीक है. You have done the procedure in, you have taken out the VNTRs, you put VNTRs from one side, आपने mother का भी डाला, father का भी डाला, child का भी डाला, ठीक है. They will start moving from this point to the other point, ठीक है, because there is anode and cathode is there, so with the help of the machine they'll start moving to the other side. But the point is that they will move if they are mother and child, or if it is father and child, ठीक है, it has to move around the same speed, okay, because अगर मान लो इसमें हमने father और child का डाला है, we have a suspect कि we have a suspect that ये father इस बच्चे का father है कि नहीं, we want to check this, ठीक है, and they are moving at different sequences or different speeds, so we can get to know that okay, maybe he is not the original father, that means उनके उसमें कुछ similarity होती है, ठीक है, in their movement, ठीक है, because क्या होएगा, variable number tandem repeats move करते हैं, ठीक है. For example father का है 2 repeats है and child has 6 repeats, so 6 repeats move करने में time लेंगे, they will take time to move, whereas 2 वाला जल्दी से पहुंच जाएगा because it's a shorter sequence, 6 is a longer sequence. So with that you can understand okay, maybe he is not the father because the child has a way longer sequence than the father, ठीक है. So now this is the whole procedure or of how we do a DNA fingerprinting. So basically we isolate the DNA, हमने बोला था this is the DNA, we took it out from one of the chromosomes, then we take the one we separate the fragments of it also, then we with the help of restriction enzyme we cut it, we took the one part, with the help of a restriction enzyme हमने कुछ part cut कर लिया, ठीक है. Then this fragmented part goes through the process of electrophoresis I showed you, ठीक है. Then this electrophoresis पे whatever we get the result, we put it on a membrane like thing or on a nylon membrane basically, ठीक है. उधर we try to study the VNTRs and then we analyze, ठीक है. So basically क्या होता है, when you put it on the you can say when you blot it on the nylon membrane ना, they start to arrange themselves, ठीक है. So वो arrangement हो जाती है of the whole sequence. Now I'll just show you, you don't need to go very deep into it, ये पूरा you can just see. So basically this is the individuals that we are testing. I'll just tell you, these are the individuals whose blood samples we have collected because we want to take out their DNA from it, ठीक है. We extracted the white blood cells and we took their DNA fragments, okay. Now we put it through a electrophoresis thing. ठीक है, we took it and we put it in the electrophoresis thing. Now it has been denatured. ठीक है, now it is transferred to a nylon membrane through southern blotting procedure. Now what is southern blotting, what is nylon membrane, you don't need to go so much deep into it. ठीक है, because वैसे भी process nobody is going to ask you कि how DNA fingerprinting is done, but in case if there is things you should know what gel electrophoresis होता है, then it is transferred to a nylon membrane. This much you can remember. ठीक है, nobody will ask you. Then after this, DNA probes, they bind themselves to the nylon membrane in a specific sequence. So this is the sequence. You can see they have bound themselves to the nylon membrane in a specific sequence. Then they are taken through a radioactive substance where they are mixed, and the film of the VNTR, this is the VNTR, the film is exposed. Okay, so this is the whole procedure. I'll just repeat the procedure again. You can just understand and just थोड़ा सा इसी को learn कर लेना. We take the blood sample. ठीक है, we extract the DNA out of it. Then we cut it through the restriction enzyme and take the fragment of the DNA. ठीक है, then this DNA fragment is separated into bands by gel electrophoresis. ठीक है, gel electrophoresis हो गया. Now these DNA bands which we had separated with the help of gel electrophoresis are transferred on a nylon membrane with the help of a southern blotting procedure. Then they prepare themselves or bind themselves on the nylon membrane in a specific sequence. So you can just skip this one also कि nylon membrane पे वो अपने आप को एक specific procedure में attach कर लेते हैं. Then with the use of a radioactive substance, the radioactive substance is mixed to this nylon membrane and the film is exposed, and that film is the VNTR. So this is the whole procedure, guys. I hope you have understood. I know it is little difficult to understand, but this much is enough. I guess you will understand. If you understand this much, nothing more you require. This much if you can understand, right? So with this we complete our chapter, and if there are any doubts, please post. I'll be happy to reply, and thank you so much, guys.